Thanks everybody for joining us again today for the Clinical Research Connections meeting. So today we have a couple of presenters to discuss some resources here on campus and some interesting things that are going on right now as well. So I do want to start with again another quick poll like we did in the first meeting, where we can just have you guys, everyone just kind of go in and identify who, which kind of their role in the research community is. So we'll give everybody just a couple of minutes to respond to this really quick, just so we have an idea of who's in attendance here in the meeting. All right, so I think we got pretty good response to that. So with that, we'll go ahead and get started here. So I want to first start by thanking Dr. Tim Dorado for coming and joining us today to discuss the Research Imaging Center, which is a great research service, research offering here on the campus that I feel like a lot of people are not necessarily aware of everything that they offer and what they can help us with. So Dr. Dorado is going to give us a quick overview of the Research Imaging Center. So I will stop sharing and let you share.
Great. Can y'all see my?
There it is.
Okay, great. And I'll start. Well, hi, everyone. I'm Tim Dorado, and I'm fairly new. I came last October, and I was recruited to lead the Research Imaging Center. We're located out in Biosciences 3 building on the ground floor, and I'll talk about what we have in there and how we're partnered with the cyclotron PET radiochemistry company, Pharmalogic. So this is just a view of our website, our main page on our website that describes our center. So our mission and values is sort of represented here on this screen. We do want to be an internationally recognized research imaging center. We can't be great at everything, so there are 3 focus areas that we think we will excel in, and that's in traumatic brain injury with the Marcus Institute. Also, we have a lot of researchers in aging or neurodegenerative diseases, and then oncology would be the 3rd area of great interest. And so we want also to be versatile and to be responsive to other molecular imaging needs of the campus. So we already have researchers in other areas such as metabolic disease that are already using the center for imaging. Our vision is to bring the state-of-the-art molecular imaging technologies, both in the MR as well as in the PET area, to bring to bear on really understanding the basis of disease and using molecular imaging to select out patients for specific therapies, and then to follow those therapies because we have such specificity for target selection. And of course, we won't be able to deliver these things without really a commitment to quality in our teamwork, in the timeliness and the innovation of the methods that we bring to bear. So I'm the director. My background is actually in radiopharmaceutical development, mainly for PET. And so I've done a lot of work with metabolic imaging in the heart as well as in cancer. More recently, I've done a lot of work with radioisotope production using cyclotrons and radio labeling from there. Dr. Bennett Chin is our medical director. Bennett has a great background, nuclear medicine radiology, and he's also done a lot of work in cancer metabolism with PET, with amino acids and others. And he's been leading several clinical trials in theranostics, and that's the combination of diagnostics and therapies. So if you have ligands that bind to tumors, you can either use a therapy isotope to deliver dose to the tumors, or you can use a diagnostic isotope for imaging. Ramesh Karki is right now our only technologist. We see that we will add to him as we go along, but since our center has been just building up, and the PET-MR, I think we're going to do our first study tomorrow, in fact, of a prostate cancer patient. We're just getting that online, and as we get busier, we'll need to add further technologists. So, but Ramesh is very skilled, and he's running both the PET-CT and the PET-MR scanner. Mark Brown is our MR physicist, and Mark has been with the, yep.
Sorry, your slides aren't advancing.
They're not. They're
no.
For me, they are.
Okay.
What are you seeing on the screen right now?
We're still seeing the first slide.
That's weird. So let me get back out. So I'm backing out. Do you see, what do you see now?
Now we're seeing the slide with Mark Brown and Ramesh Karki.
But if you can put it on projector view, that might be great. I think that might be the hang up.
View. Slideshow.
Yes.
I don't see projector view.
You should just be able to select from
slide. Yeah. Can you see that slide very well?
I mean, yeah, we can see the slide. It's usually a little bit larger and easier to see when it's in presentation mode. That works like that.
Yeah. That works. Okay, let's go with that. So anyway, Mark Brown is also working at the Brain Imaging Center, or BIC, and he's sharing time with us at the RIC. And he's, of course, an expert in MR physics, including helping us to get the new sequences up on the scanner and troubleshooting things there. Our other staff, shown here on this slide, Abul Lakhani is our Director of Finance for radiology. He's really working with the finances of the center. Dan Lopez Panagua is an image an analyst. So he's working with analyzing the images, both MR and the PET, to get the data out of, the relevant indices of function out of the data. Mike Solosky is our nuclear medicine physicist. So he's really responsible for our PET-CT for quality assurance of that camera. And then we have an on-site GE scientist, Dan Litweiler. And Dan's a great asset to us. He's with us for at least 3 years, and we hope he'll be with us more than that. And he's an MR physicist, and so he's also working with Ramesh to bring up the PET-MR and get all the sequences right when we go into scanning. Ann Scherzinger is almost retired, but Ann is with us until she is retired very soon. And she's worked in a variety of quantitative imaging analysis areas. And then our receptionist and scheduler is Jessica Ruvalcaba. So our organizational chart is kind of shown here. I hope you can read some of it. Basically, we are overseen by the School of Medicine. Dr. Peter Buttrick in the School of Medicine Dean's office is our chair. And then we answer to them. From the radiology side, it's administered through our chair, Dr. Chip Dodd and Mahol. And then there's me, and Dr. Bennett Chin is our medical
I think he might have gotten cut off. Is anybody else having issues?
Yeah. Dr. Dorado, are you there? Okay, well, we'll give him just a minute to see if he can get back on. Hopefully it's just a quick blip in his internet maybe, and he can jump back onto the meeting. Okay, well, while we're waiting on to see if Dr. Dorado can rejoin us, we'll go ahead and just move on to our next presenter. And then hopefully we can have him come back once we get through that. So our next presenter is Leah Emmerich. Leah is a is new-ish to campus. I'm sure many of you have not had the opportunity to meet her yet, but she is a process improvement specialist here on campus. And she is actually within the Office of the Vice Chancellor for Research. And she's going to talk to us about some process improvement work that she's got going on and about the process improvement process. So Leah, I will turn it over to you.
Hi, everyone. I'm going to take a second here to share my slides, and then I'll dive in. I have about 15 minutes planned to talk with you. So hopefully we can get back to Tim when I wrap up. So let me share. You able to see my slides okay?
Yes, we can see them.
Let's see if I can do the presentation mode here. How's that?
Looks good, thanks.
So welcome everyone, good afternoon. I'm really excited to have a chance to speak with you all about my new position. I was excited last time to see some familiar names. I'm a returning CU Anschutz employee. So I noticed a lot of old friends and colleagues that were joining this conversation the last time we met. So happy to be here to talk to you about my new work. For those of you who don't know me, I started back at the Anschutz campus back in 2007. I worked in the Barbara Davis Center for a number of years before moving on to the OBGYN department. So I started as a PRA and worked my way up to senior before I left. I worked in applied public health for some time and wound up at Denver Health, where I was formally trained in process improvement methodology, was able to apply that both at the local public health level, as well as when I left, I was a senior lean facilitator at Denver Health and got to apply these skills to a number of settings. So the role that I'm in now, this I'm just sharing a little bit about the objectives of my position. I was hired the end of May this year, and Dr. Flagg really has the goal with my position to help implement a quality process improvement program within the Office of the Vice Chancellor for Research that will help drive improvements in both our research and operational outcomes at the Anschutz campus. So some of my initial responsibilities will be supporting process optimization within the OVCR. In that space, I'll be partnering with leaders in different areas to help identify and map what are our current processes and services, who are our customers, how can we look at that from a process improvement standpoint, and really engage teams in using that methodology to drive positive change. So I'm very excited to already have been diving into that space with some of you. Just to share a little bit more about the why, it's really critical for our identity as a research institution to ensure that we're optimizing our effectiveness, that we're staying competitive in the market of research. And also, you know, in a post-pandemic environment, many organizations are really thinking about and reconsidering how our work is done and rethinking how innovation might become more part of our everyday work. A lot of organizations bring in quality improvement practitioners as consultants, or they may hire someone on full-time. I feel fortunate that this was put together as a full-time role that I can really engage and learn more about the work that folks are doing in Anschutz to support those long-term changes. So a culture of continuous improvement is something that I'm trying to help establish within the Office of the Vice Chancellor for research. Ultimately, the objective is to start empowering staff that are working in those support roles to think in innovative ways about the business of research, empowering them to think about what are the things that don't always go so well in our day-to-day work that we can find ways to experiment and make positive changes. It's really critical for us to allow staff to be engaged in that, because those of you who are doing the work every day are the ones who often can have the most innovative ideas for a better way. A lot of practitioners will call this a lean journey that organizations take as they're starting to think about how do we shift from the way we've always done things into more of a mindset of continuous improvement and really building those methods and principles into their daily work. So I'm going to provide a little bit of an overview of what lean methodology is, a little bit about the services that I'm going to be providing within the OVCR. I won't have a ton of time for questions, but I'll leave my contact information at the end. So here's just a high level overview of the QI methodology. So lean is a systematic approach to continuous improvement. It uses guiding principles, it uses tools to help us think about how can we maximize value for our customer, and how can we do that at a minimum cost to the organization. Lean really came out of Toyota and their manufacturing. They've applied these principles for years and years, and has since this methodology has been applied in many cases in hospital systems like Denver Health with different service providers. It's gone well beyond manufacturing. Many institutions of higher education are engaged in lean practices as well. So really widespread. A lot of people are starting to think about this now. And the basic goals are to eliminate waste, things that don't add value to the customer, reduce costs so that we're using our funds with stewardship and making the most of what resources we do have, and improving quality. So improving the quality of those services or outputs that we provide. Just want to bust a few myths about lean. It's not an acronym. It is not just about tools, although some people, that's a great way to start a lean journey, is to learn some of the tools and how to apply them to your work. It's not about cutting staff. It's not intended to be punitive in any way. It's not only useful in manufacturing. It's not actually a program. It's not a substitute for leadership. It's not a silver bullet, and it's not the job of just one person. And I kind of emphasize that because although I've been hired to help support and coach others in adopting this continuous improvement methodology, I can't do that as one person in this role. I hope to be able to share some of those principles and tools with others so that we can really build as a community of problem solvers and help drive the higher level vision for research at our medical campus. So 2 of the pillars of lean, first is respect every individual. This is the belief that our staff is the organization's best resource for quality improvement and innovation. And the second is continuous improvement. So the idea that we want to create a vision for an ideal state, knowing that we never will actually reach a state of perfection, but the idea that we can always be continuously improving as we advance in our work. Things shouldn't stay stagnant. We should always be thinking about those ways we can incrementally improve. Leading with humility is really an enabling principle. We can't have respect for every individual or a culture of continuous improvement without that humility. So really coming into the space, knowing that improvement is only possible when we're willing to acknowledge vulnerability and just come to the table together to problem solve and really have a shared vision for the future. So a little bit more about what lean is. It is about identifying the processes that we do, the things we do every day, understanding what services or what offerings we deliver. It is about how we behave in the workplace, our management practices, how we work together as teams, how we use respect for people and continuous improvement in our daily work. And there are numerous tools that can be applied to different problem solving situations to help guide teams and leaders through those continuous improvement exercises. If you look into lean, the typical first principles of lean start with defining value. So understanding who your customers are, how do they define value, mapping the value stream. Those are the steps that a customer takes when they're working through your work. 3 is creating flow. So making sure that there aren't a lot of backlogs or waiting times in the services we provide. Establishing a pull system where customers can ask for or request things in real time without much delay. And again, I mentioned pursuing perfection, although we know we won't ever reach that ideal state, just having that desire to continuously improve and work on those incremental changes that can make us a little bit better every day. So lean thinking defines value as providing benefit to the customer. Anything else is considered waste or non-value added activities. It may sound extreme, but really understanding that customer value, understanding the things that help your teams operate in a way that's respectful to them so that there's not a lot of back and forth in the work that they're doing. That's all part of shifting into that mindset of lean thinking. Lean in practice, as you get into the problem solving space, really just uses the scientific method. So it's taking an initial perception of a problem. It's analyzing deeply the current state, diving into root causes that may be contributing to those less than ideal outcomes, and then working together to define a target state. We act, make plans, do experiments, and measure to see if what we've designed as a target state, have we really moved the needle on the outcome? So in lean, we talk about the PDA cycle. This is just plan, do, study, act. So we're planning, looking into that current state. We're doing experiments and making small incremental changes to see if things are changing in a positive way. And as we study them, sometimes our ideas, our hypotheses for what a better way is won't always be the right answer. And so we go back to the planning phase. But this cycle is something that teams can engage in continuously at different scales. We can do this at a high level. We can do this at an individual level, even with our own work. Step 4, acting, is really once we've identified a change that has shown improvements, that we operationalize that and track it over time. And that's part of the process as well. So again, dreaming of the ideal state, the ramp of improvement would be over time, we're doing those incremental improvements, making experiments, creating operational standard work that helps hold those improvements in place. And we're slowly moving up this ramp from how things are today, where we might not be giving the highest value to our customers, and over time, we're getting a little bit closer to this ideal state. So that was a lot of very high level information about lean methodology. I'm just going to give you a little bit of an overview of some of the pieces I'm hoping to support within the OVCR. And I'll provide, again, my contact information if there's questions or if anyone wants to connect. So as I'm thinking and working to implement a culture of continuous improvement within the OVCR, a balanced approach is really key. Part of it's creating capability and teaching others about those principles and tools, allowing them to learn how that can be applied to their work. So that's part of creating capability. Aligning work to strategic plans is really, as our on-shoots strategic plan is coming out, each team really thinking about how does our work tie into those high-level goals? How can we as study teams or we as support teams, how can we drive the mission that we have to make our research the best in the country or the best in the world? How do we align our daily work to those higher level strategic plans is definitely part of the puzzle. And then helping others learn how to manage and continuously improve the processes that they're part of, whether it's a frontline team member or frontline leader or higher level leaders. I'm hoping to be able to help others apply these principles into their daily practices. So some of the services within OVCR that I'm working on and am engaged already in a handful of improvement projects, but it's really supporting teams, guiding them through that PDA cycle, helping guide them through with fidelity, the process improvement methodology, providing training and education, providing coaching to those who really want to learn how to apply some of these skills and tools to their own work. And then again, strategic support, thinking about how can we align from the top down and also from the bottom up. Because I'm only a one-person team, I can't provide these services to everyone by myself all the time. If you do have some ideas that have come to mind, projects or things that you're really excited to share, you're welcome to email me, and I have an intake process for really considering work that can help drive our research goals. But I want to point you also to some resources that are already in Skillport. So if you look under the instructor-led trainings, there's 3 continuous improvement courses that are already well established. And the person who put these into place actually got their training at Denver Health originally. So it's very well aligned with what I hope to share with others. But fundamentals of continuous improvement, intro to continuous improvement tools, and continuous improvement understanding the current state are 3 instructor-led trainings that Debbie Lammers provides through the Office of Learning and Development. These are really great foundational courses. And if you are looking or wanting a whole team to go through one of these, you're welcome to email me or Debbie and just let us know, hey, we want to learn a little bit more, and this is a great place to start. I'm going to leave my email here and I can pop it into the chat as well. But if you do have questions about the work that I'm doing, you're welcome to reach out to me. What I would ask, I know some of you are in supportive roles within OVCR. Others of you are probably investigators or other research team roles. If there are things that are really challenging you in your daily work and you want to just shoot an idea out, you're also welcome to email me with ideas that you have for improving things. So I'll put my email into the chat, but want to make sure we have time to go back to our other presentation. So I'm going to stop sharing. And then Aaron, I'm not sure if we have time for questions at the end. You could come back to me. If you want to pop back to the other presentation, you let me know.
Yeah. So thanks, Leah, for for your presentation. Does anyone have any questions for Leah? If you want to go ahead and you can unmute and ask your questions, or feel free to type anything you might want to into the chat as well. All right, if no one has any questions right now for Leah, definitely feel free to reach out via directly to Leah via her email, or there's certainly the Clinical Research Connections email that you can send an email to, and we can make sure that Leah gets those questions. All right, so it looks like Tim is back, and so we'll turn the presentation back over to him and let him finish up his presentation.
Thanks, Aaron, and I apologize. My computer is a little slow or something and it dropped. But anyway, I think I was about here in the presentation. I was going to go through what our specific imaging modalities are, starting with the PET-MR. So we have a state-of-the-art GE PET-MR machine, and that combines the PET detectors, which sit inside the MR gantry. You can't see them here with the cover on, but they're right as close as they can be to the patient to give good sensitivity and of imaging of the PET photons that come out of the body. So PET gives you biochemical function, and the MR is used for anatomical fusion of the images to the PET. MR also has some functional measurements such as diffusion, which measures diffusion of water in the body. Tractology, which sort of connects areas, say, of the brain, of one part of the brain to the other part of the brain. And then there are other things like you can do contrast injection with MR to get what's called DCE MR for looking at blood flow to the tissue. Some of the areas of applications here, of course, neurology, imaging of the brain in health and disease, traumatic brain injury. So we have a major collaboration with the Marcus Institute for TBI, and they're doing a lot of imaging in veterans, as well as in sports injuries and other types of brain injuries, psychiatry, oncology, and we have metabolic imaging, of course, with diabetes and obesity and so forth. On the PET-CT side, we have a Philips time of flight machine. And in this case, the CT gantry is separated from the PET gantry. You see the CT gantry here in front, and the PET is in the back. And the scanner table, which holds the patient, goes in between these two. It gives a little bit more open environment to the scanner than what you saw with the PET-MR, but then allows the patient body to be moved between them. PET, of course, again, gives you biochemical function, and CT is mainly for anatomical mapping with fusion with the PET scan. The CT scans are very rapid, only takes about 2 minutes for the whole body. After acquiring the images, we want to extract information from those images, be it the biochemical information from PET or the anatomical information from the MR or CT. And so here are some of the parameters we get off the imaging. And Dan Lopez is our leader in this area. So you could be, looking at tracking of lesions, looking at volumetrics. Say you have an area of the brain that's affected by disease, and you want to follow that over time with the volume. Or fMRI, which is functional MRI. Parts of the brain may light up as you think about something. And other areas here. So we also have a group that will be analyzing the images and working with the PIs to define what the best type of approach for analysis would be, as well as doing the image analysis. The other option is that if the PI has that area in their own research group of analysis, we would then just basically hand over the images for them to do the analysis in their own group. Like I said, we're partnered with Pharmalogic, which is the company that owns and operates the cyclotron that's next door to us. So they are literally next door to us. And that's great because some of these short-lived isotopes, you don't want to lose them by radioactive decay in the transport. So we're very close. We can do even as short as oxygen-15, which has a 2-minute half-life, very short. And so we can even pipe that over directly to the scanner rooms and look at oxygen metabolism, oxygen uptake. The isotopes can go all the way out to multiple days in half-life as well. The PET cyclotron is a GE cyclotron, 16 MeV. It's very flexible, can make a variety of isotopes. And Pharmalogic has not only the provision of routine clinical PET tracers, but our research tracers. So we're partnered directly with them to provide all of the radiopharmaceuticals for us. As I said, part of my background is in radiopharmaceuticals, so I'm serving as a consultant over to Pharmalogic as well. Some of the tracers that we use are shown down here at the bottom. Our workhorse FDG, fluorodeoxyglucose, a sugar molecule taken up highly by cancers. A metabolic tracer we use a lot is C11 acetate. It's an oxidative metabolism probe. PIB is an amyloid imaging probe for neurodegenerative disease like Alzheimer's. N13 ammonia is for myocardial blood flow. Dotatate analog is taken up by a somatostatin receptor in tumors, such as neuroendocrine tumors. And PSMA is a compound that binds to prostate cancers. FET is a amino acid, which would be useful for brain tumor imaging because the normal uptake in the normal brain is very low of this amino acid, so it really gives a good contrast in the tumor. These are just a few examples of tracers, and there's ongoing development. So the good thing about is if you have a sponsor that has a biological target and has some idea of what type of molecule could hit that target, we can work with that investigator and talk about bringing up novel radiopharmaceuticals for their biological targets. So that's what we have here. Also, I wanted to mention that Pharmalogic has a significant infrastructure. So they have 45 to 50 radiopharmacies across the US, Canada. I think they're in Puerto Rico as well. And they have a centralized regulatory arm of their company that oversees the regulatory practice in each of their sites, such as Denver. Denver is the first site with the cyclotron, but I think they have plans to bring up 6 or 7 by the end of next year. So they're quite on the growth curve as a company. So that gives you an idea of who they are. And then lastly, we have our contacts here for our center. Our scheduler is Jessica here. And for any scientific questions, you can talk to me. So I'll open it up for questions, and thank you very much.
All right, does anyone have any questions for Tim? No. All right. If anyone does have any questions, again, you can certainly feel free to reach out via the Clinical Research Connections email, or obviously Tim gave you his contact information there as well. If you have any questions you'd like to ask directly to him. So I do want to say thanks to both Tim and to Leah for coming and presenting today. I do want to just remind you guys all that this meeting, while we do have several topics, ideas for topics that we'd like to present during these meetings, these monthly meetings, there are obviously we want to make these meetings work for you all. And so if you have any topics that you feel like you would like to discuss, please feel free to, you can either drop them in the chat now, or certainly send an email again to the Clinical Research Connections email address as well. So we did get a couple of recommendations during the last meeting, one regarding an overview of eReg and how that works and how we plan to use that. We are working on getting a presentation scheduled to do an overview for that. And then there were a couple of other questions regarding resources and mentoring for new coordinators on campus as well. So those were a couple of ideas that were presented last time from the group. If anyone does have any questions, again, feel free to drop them in the chat now, or send an email to clinicalresearchconnections@cuanschutz.edu. So with that, that's everything that we had planned for the meeting today. So I'll open it up to anybody who would like to just have some discussion now. Does anyone have any questions or topics they'd like to bring up and address? All right. If not, then thanks everybody for your time today and for joining us for this meeting. I hope you all have a great week, and we'll see you next month. Thanks.
Thanks, Erin. Thanks, everybody.